Welcome to today's Explainer. I'm really thrilled to have you here. Look, if you've spent any time online or paying attention to the wellness world lately, you've probably heard some incredible, almost magical claims about microdosing. Silicon Valley executives absolutely swear by it, influencers are out there practically selling schedules for it, and millions are convinced it's a total game changer for their brains. But here's what we're going to do today. We are going to completely separate the massive cultural hype of LSD microdosing from the actual hard scientific evidence. We're pulling directly from a comprehensive deep dive published in the OOTW journal to see what actually happens when you put these big claims under the microscope of tightly controlled laboratory settings.
So, that brings us right to the million-dollar question. Does a tiny, nearly invisible chrome of LSD taken every few days genuinely alter your neurochemistry for the better, or is it entirely possible that it's all in your head? What do the randomized control trials actually prove? We're doing a reality check today, and honestly, the scientific truth is way more fascinating and a lot more sobering than either the hardcore believers or the skeptical debunkers will have you believe. Okay, let's dive into this and get our baseline straight. What exactly is a microdose? Well, a true microdose is defined as a sub-perceptual dose. We're talking roughly 5 to 20 micrograms of LSD. To put that into perspective, that's only about 1/10 of a standard recreational dose. The whole idea here is that this tiny sliver of a compound is small enough that you shouldn't feel obviously high or altered, but it's supposedly just large enough to subtly nudge your mood, sharpen your focus, or unlock a bit of quiet creativity. Now, how are people actually taking this? You can't just take it randomly if you want the supposed benefits, right? Enter the famous Fadiman protocol, which was popularized by psychologist James Fadiman back in 2011. It works on a simple 3-day cycle. Step 1 is your dosing day. Step 2 is an off day to let the effects integrate into your system. And step 3 is another off day, specifically intended to blunt the rapid tolerance that your body naturally builds to LSD. You repeat this for a few weeks, then pause. But here's a crucial takeaway for this explainer. This widely used, very official-sounding schedule, it's basically a folk protocol. It was refined entirely through anecdotes and user surveys, absolutely not by rigorous clinical dose-finding trials. And well, relying on those anecdotes leads us to a massive plot twist in the scientific literature. When people microdose in uncontrolled surveys, they overwhelmingly say it works. But researchers knew they had to control for belief. So a team led by Balazs Sigeti designed a landmark 2021 self-blinding citizen science study. Participants set up their own capsules inside QR-coded envelopes, meaning nobody knew who got the drug and who got an empty capsule until the very end. The results from the roughly 190 people who finished, the microdose group saw significant improvements in well-being, mood, and life satisfaction. But get this. The placebo group saw the exact same significant improvements on the main outcomes. The empty capsule worked literally just as well as the drug. What that study revealed is that the real star of the microdosing show is expectancy. It's the placebo effect just doing honest, heavy lifting work. You see, when people believed they might be microdosing, their mood improved, regardless of what they actually swallowed. A later 2023 reanalysis showed that when people guessed they took the drug, which is what we call broken blinding or activated expectancy, it manufactured a completely false drug signal. It's undeniably ironic and maybe a bit humbling to realize people were walking around feeling like absolute creative geniuses when they were essentially powered by a completely empty capsule and the sheer force of their own expectations.
And this brilliantly illustrates one of the most consistent patterns in all of microdosing research, the huge gap between feeling and reality. Subjectively, people rate themselves as having higher creativity, sharper focus, and better well-being. But when researchers actually measure objective performance on cognitive or creativity tasks, the needle barely moves. The subjective feeling tracks perfectly with whether people believe they took a real dose, totally independent of their actual objective output. It's just a powerful reminder of how human psychology works. Feeling more creative and actually being more creative are simply not the same thing.
But what happens if we strip away the self-reporting entirely and just run a highly controlled, repeated dose, randomized trial? Well, Harriet DeWitt's research group in Chicago did just that. They had 80 healthy volunteers take either 10 micrograms of LSD or a placebo every 3 days for 6 weeks. And their summary paints a pretty sobering reality. LSD produced transient mood lifts on dosing days, but no lasting change to overall mood or cognition. An acute, temporary blip in your mood on a Tuesday just doesn't equal a durable, life-changing benefit. On top of that, the study noted that anxiety was actually the most common adverse event reported.
All right, moving on to Section 1, the real pharmacology, not an inert crumb. Because To be fair, low-dose LSD is not just an inert crumb doing absolutely nothing to the human body. In tightly controlled environments, acute microdoses cause real, measurable, mostly acute physiological shifts. It alters blood pressure, changes sleep patterns, shifts neural connectivity, alters how you perceive time, and even changes your EEG readings, specifically reducing certain oscillatory rhythms in your brain. So the brain absolutely notices the drug is there. But again, these effects are modest, highly dose-dependent, and pretty fragile.
Now, what's really fascinating about this number is this specific threshold, 20 micrograms. The controlled lab work shows us that 5 micrograms essentially does nothing. But right around that 10 to 20 microgram mark, the effects become distinctly noticeable. In fact, in one striking study, a 20 microgram dose actually increased tolerance to pain, specifically cold pressor pain, to a degree that authors compared to opioids. But this only happens right at that 20 microgram threshold, which is exactly where mild psychoactivity, the feeling of actually being slightly altered, begins.
Which brings us to Section 2, Safety and the Headlines, where we need to make some crucial distinctions.
So, the crucial point is, we have to stop conflating the headlines. The news loves to run stories saying things like LSD shows therapeutic promise, but they are constantly mixing up two entirely different things. On one hand, we have microdosing, taking 5 to 20 micrograms frequently over months with unproven durable benefits. On the other hand, we have actual clinical successes, like the MM120 trial for generalized anxiety that recently earned an FDA breakthrough therapy designation. That trial used a single, full therapeutic dose of about 100 micrograms paired with heavy psychological support to achieve durable anxiety reduction. That is the exact opposite of microdosing. We've got to stop confusing trendy, frequent at-home habits with massive, single-dose supported clinical trials.
And the reason this frequent dosing habit matters so much is safety. While short-term studies in healthy adults show it's generally well tolerated, there's a serious mechanistic worry when it comes to chronic use. LSD doesn't just bind to the brain receptors we want, it also binds to the 5-HT2B receptor. Why does this matter? Well, chronic frequent activation of that exact same 5-HT2B pathway is the known mechanism behind heart valve disease. It's the very same pathway that got the infamous fen-phen diet drugs pulled from the market. Now, this is a theoretical risk for microdosers, but because the whole protocol involves frequent, repeated dosing over months or years, researchers have heavily flagged this heart valve risk. Currently, there is a total lack of long-term human safety data to say one way or the other. It's an unresolved, unstudied question.
Which leaves us with this final, incredibly provocative thought to wrap up our explainer today. We've looked at the data. The big, durable, therapeutic claims are still waiting on big trials, and right now, the evidence points heavily toward expectancy. But if the well-being and the mood lifts people report are mostly driven by placebo, yet the subjective feeling of improvement is completely real to them, does it ultimately matter if it's just in your head? The sheer, unadulterated power of human belief remains undeniably fascinating. Thanks for joining me to walk through the science behind the hype. Keep asking the hard questions and keep learning.